right. My name is Sten Vermund. I am happy to serve as the Chief Medical Officer of the Global Virus Network, and very happy to introduce my friend Fernando Spilki. His topic today is going to be virus genomic surveillance and One Health, diving into the Brazilian virosphere. Obviously, Brazil is one of the most fascinating countries on the planet Earth, with tremendous diversity in ecosystems and therefore tremendous opportunities for emerging pathogens and re-emerging pathogens. We also have the phenomenon in Brazil of the Amazon being increasingly populated, so that we're bringing humans in contact with animals, including insects, at rates that are unprecedented. Dr. Spilki is a full professor at Feevale University in Brazil, and he is also the vice rector for research. He has a very distinguished career, many titles and many awards. And rather than go into all of these, I just want for you to know that his focus on viruses is both in domestic and wild animals, as a veterinarian and as a PhD, and emerging zoonotic viruses, so those that are of human relevance. He worked on SARS-CoV-2 recently, as many of us did, but he's also looked at viral contaminants of food and water. So I'm extremely curious about what he's going to talk about today, and I don't want to take any more of his time. So Fernando, it's all yours.
Oh, Dr. Sam, thank you very much for the introduction, and thank you very much to GVN for the opportunity to be here and present some of our work. Actually, one of the things that we lost over the years was focus. Actually, we are now focused in many things, and I will show you a glimpse of all the activities that we have. And the main objective of my presentation is to show you what we call the Brazilian a national network for virus genomic surveillance and One Health. That's the major project that is ongoing right now, and I have the honor to coordinate this. I will tell you something about the origins of this project, whys and the do's of the project, and as well as some of the results that we have already have in terms of what could be this tremendous virosphere that we could see here in Brazil, and the idea or the motivation, the primary motivation for our work is the tremendous biodiversity that we have here. Brazil is #1 in terms of biodiversity in many different taxonomic groups, and actually just an example, we are #1 or in terms of biodiversity of bats, and everybody in this room know how bats are important species in terms of what could be the viruses that they could harbor, and but they're not only bats. We have a huge diversity in terms of marsupials and any kind of mammals and many other types of living beings. And biodiversity is actually good. It's a tremendous treasure that Brazil has. But the problem is that this biodiversity is connected, unfortunately, to a huge environmental impact nowadays. And Brazil, of course, we are well known by the Amazon, and Amazon is by far the most impacted biome that we have nowadays. Although the mega diversity that we have there, the problem is that the expansion, especially of mining and agriculture and disorganized urbanization, these are problems that are, like Dr. Stem mentioned, making that we could face or to get in very close contact with other species that could harbor viruses that are there and sometimes could jump to humans, but not only these. The thing is that we are disrupting nowadays a lot of the ecological uh, 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 imbalance that we have in Amazon that is huge. And the thing is that most of these species are now being threatened by the presence of humans and the disruption of the local landscapes. But there is not only Amazon, we have at least other four main biomes in Brazil, and most of them are now hugely impacted, although they have a huge biodiversity that was meant to be preserved and conserved. Sometimes, especially the expansion of agriculture and urbanization in the other regions in Brazil, are major threats for the conservation of these species. And of course, Brazil is in the center of the problems that are coming with climatic changes as well. I live in Rio Grande do Sul, which is the southernmost part of Brazil, the southernmost state of the country. And as you know, in 2 years ago, we had these huge floods here, and that had an impact even in the conservation of species, and that had direct effects, for example, in the transmission of dengue that year. And thus, we are seeing these effects of environmental changes in a daily basis in Brazil. As you know, before the pandemics, I always said that many of the factors that could drive to a new pandemics were known to many of us. We teach these in classes, but during the pandemics, it was like a nightmare class. We saw how a virus can jump from nature to humans. And one of the things that I think that we all have learned is that we have to monitor other species, environmental matrices, and foods for the presence of potential new viruses. It is difficult to say, actually. I think that before the pandemics, we had some kind of certainty that these and these species had to be observed, and these and these type of foods or environmental matrices have to be monitored. I don't think that we have this huge amount of certainty nowadays, but at least we have to look to most of these samples in a way that at least could give us a hint of what could be the next virus that could jump to humans or to domestic species. And that's the thing that we are trying to do in a way. Of course, the holy grail of all these searches and surveys that we are conducting is to predict the possibility that a given zoonotic virus could reach human beings. And sometimes this is not only the justification of our work, but it is also the main source of critics that we receive. Because sometimes people can say, and they are not totally wrong, that well, you were looking at the nature and you did not see the viruses that were coming over the last 30 years before they reach us. But here and there, we could find these viruses. And then we had a chance of, like, it was like an assay of what could happen in terms of protection of species that are nearby us. We saw the presence of this virus, virus, in the water that was collected in swine farms here in Rio Grande do Sul. At the middle of the year, I think that it was 2016, and by the end of the year, and the virus was not detected at that time in animals, but at the end of the year, we saw not only the introduction of the virus in these farms, and we had the chance to detect the virus in animals, but it was already causing disease 6 months after we have detected the genome of the virus in water. So it might be not as important as an assay for pandemics, but it shows that it is possible. You could look for nature, and then thereafter you could see the virus causing disease in a given important species. Actually, what happened in Brazil is that during the Zika years, we have developed the skills and the infrastructure in Brazil to do some sort of genomic surveillance, but it was not enough for this purpose. But during the pandemics, and although it was terrible to all of our countries, especially for Brazil, there are some legacies that lasted. And one of them is that we had the chance by an initiative of the Ministry of Science and Technology, although we had these huge problems with the Brazilian government in terms of how to deal with the pandemics at that time, the Brazilian Ministry of Science and Technology had the initiative to promote the formation of different networks to establish collaboration between Brazilian universities and centers of investigation for some focal important targets during the tackling of the pandemics. And one of them was the so-called Coronaomic. It was a network for virus, SARS-CoV-2 genomic surveillance. Our idea was to share expertise and to do capacity building in terms of genomic surveillance during the time of the pandemics, and also help the Brazilian Minister of Health and other local authorities in what regards to the genomic characterization of the virus. And that was very interesting because it allowed us an opportunity to join many groups that, although we recognized ourselves as a virologist, and we had some focal or dual collaborations, at that time we had a chance to really collaborate as a network, and that was very important. That was new in terms of Brazilian virology. Together with that, we had this Previr network that was not meant for genomic surveillance of specifically SARS-CoV-2, but it was for possible emerging viruses that could be circulating also in the Brazilian fauna. And the thing is that actually the networks were somehow connected, but we learned a lot also in that time of how to perceive or how to monitor this kind of viruses in nature. And in the end of this process, what we did was to check with the most close partners what could we do for the Brazilian society for the next years. And that was the idea of the basic conception of this INCT-One Health, or INCT-One. That's a Brazilian national network for virus genomic surveillance and One Health. And our main idea was to contribute on the genomic surveillance and the aftermath that could come from that, the isolation of viruses, characterization of the zone of reputation, and all these of viruses that could be circulating in wildlife, in domestic animals. That is a new subject in this topic, at least in Brazil, and also of search of what could we contribute in terms of viruses that are already circulating in humans. Our main focus is on the capacity building, training of human resources that could really do all this kind of stuff, studies from sampling to the analysis of data, and to establish actually the basis of a collaborative network that could be even more expanded over the years. These are the folks that are participating in our network. I think that some of these faces are well known to you. Mauricio Nogueira is the vice coordinator of the network, and we have focal points of this network in both in south of Brazil, the southeastern part of Brazil, and center-western part of Brazil. Of course, we have the northeast and we have the Amazon outside the network nowadays, but we established some collaborations with people from these different centers. And one of the most important that I could see here, of course, there are many others, is with the Vivera project. These guys are doing an spectacular work at the Amazon basin, and they look for what is happening in the villages, the small villages nearby the borders of Brazil with different countries in the Amazon context. And they do not only get samples from humans and domestic animals, but also from humans animals that live in the forest, animals that were hunted by the indigenous people, and every of these samples are being also driven to our laboratories at the INCT-One to check what kind of viruses are there. And this is the type of collaborations that we have here. And in terms of international collaboration, we have many other collaborations, but two of them that were very important and are very constant collaborations for us are these with the UTMB at Galveston, and also with the Charité Institute in Berlin, and particularly in UTMB. We are very thankful for the Nikos Vasilakis group, which is in close contact, especially with Mauricio Nogueira, doing a lot of work in arboviruses, and also the Charité Institute in Berlin, especially with Jan Felix Drexler, which is a very close collaborate with many of our groups here in Brazil. And one of the major sponsors at the beginning of this network, especially with the transfer of expertise of how to conduct a study like this, was the UK Health Security Agency. But of course, we are open for collaboration with any other groups from any part of the world. What kind of study we do? Actually, at the very beginning, what we do is the search, is the survey, and our idea is to use the more promiscuous protocol that could be used in terms of metagenomic for to look for the virums that are in different types of biological samples and also in environmental matrices. As many of you know, the way in which we extract the RNA and DNA of the samples, and the way in which we do the library preparation will input a lot of bias or not. And in this particular paper, what we done in this study, what we done was to look what kind of protocol could give us the more diverse findings. And we were very happy that at least in two of the four protocols, what we found was very good in terms of the diversity of viruses from RNA to DNA viruses, and the number of genera and families that we were able at least to see as hits, or even partial or whole genomes that we have found in different type of samples. And that was soon applied to different studies. This is one that was published last year. It is here with the fauna from Rio Grande do Sul. We have searched for viruses both in different terrestrial animals and in bats. in a very populated area, hugely impacted by the occupation or disorganized urbanization, entering different landscapes and forests in the southern part of Brazil. And the thing is that we look for different types of mammals. And one of the mammals that become at this very beginning as a champion for us was opossums. It's amazing the amount of different viruses and families that you could find in them. So it is we had some studies in the past showing that opossums could be very prone for spillback from viruses that are circulating in human population that could reach them. And in the end, we saw that it's not only human viruses, but also viruses from other species that could reach these animals. And this is very, very interesting in terms of what are we trying to do, that is to monitor nature wildlife for the viruses. And we also we are always looking for anything in any kind of animal. And one of the things that we have done is collaboration with different groups. This is a group from the Northeast part of Brazil in which we were able to look for the samples that they had in different types of rodents, and we were able to find this hepatitis C that was published in last year. And another interesting finding that we had is some sort of preliminary, but it is now being still being studied. It is about the situation of a possible new type of hepatitis E in capybara. We do not know about the zoonotic potential of this virus, but it is interesting to see that it is quite different from the type of hepatitis E4 that is circulating in other species. And it is also impressive that after the discovery of this in metagenomics, we did, in collaboration with people from the Fiocruz Recife and the Charité Institute, we did PCR and also serological survey in many other samples of capybara, and it is impressive that this virus is not rare. It is also actually very common. More than 58% of all animals tested were harboring this virus. So it seems that it is something that is very present in capybaras, and capybaras is nowadays like a plague in Brazil. They are everywhere. And of course, we do a lot of survey in bats. And from bats, I can tell you that we have the normal stuff. Alpha coronaviruses everywhere. We could see this not only in the southern part of Brazil, but there are also studies from Helena Lagen and her group showing the huge amount of alpha coronaviruses that could be seen in the southeastern part. There are many other studies by Edson Burigon and other collaborators in other parts of the country. And we have already published some of these studies. There are some viruses that are not so nice, like we saw other alpha coronaviruses that are actually infecting more than one species of bats, and these are very present. So nowadays we are trying to give more to do more studies in about the biological characterization of these viruses as well. And we have some viruses that are quite worrying, like this beta coronavirus it's quite close to older MERS coronaviruses that were detected before. It is circulating not only in Rio Grande do Sul, but also in Santa Catarina. We saw this virus circulating also in different settings, and it is impressive that that's not the first MERS-like coronavirus that is circulating in Brazilian bats. There are another genomes that are circulating in other bats from the southern part of Brazil, And it was described by another group in the northeastern part of Brazil, also another MERS coronavirus that it's quite different from this one. So we have, as you see, a kind of diversity, genomic diversity of MERS-like viruses that are there, that are outside circulating in at least three different species of bats here. Another target that we have is about H5N1, and specifically in the southern part of Brazil, we have this huge problem that there was a major outbreak. I wouldn't say that this is an isolated outbreak. Actually, at least for the last 2 years, this virus is circulating continuously in different colonies of sea lions in the southern part of Brazil. And what we are seeing that was already described in other portions of the country is that the virus is evolving all the time. It is changing. And it is actually causing a very severe disease in these animals. With metagenomics, we did not show only that the H5N1 was a source of disease in these animals, but we also saw other viruses like picornaviruses and parvoviruses also infecting these animals. So it's kind of interesting because we are looking for a given virus that worries us, but with the aid of metagenomics and other techniques that come further, we were able also to see viruses that are a real problem in terms of the health of these animals and the conservation of these species as well. And we also actually did all the characterizations, and we are still monitoring about the evolution of this virus, which had, as I said, a huge impact on the sea lions colonies here. And Brazil, outside the huge biodiversity and more than 210 million peoples that are living here, we have a lot of domestic animals, and animal husbandry is very important for the Brazilian economy. We are like the one of the greatest exporters of beef, meat, pork, and poultry meat as well. So this is kind of important for us. And this could be actually important as well as a source of jumping of different viruses to human beings. So we did a lot of work in the past, and we are continuing to do that. Is like an example is about the monitoring of viruses with zoonotic potential in pork or other meat products. And we are also applying metagenomics to check what is circulating and what circulated in the past in Brazilian animal herds. Like this is only an example of a work that is being conducted right now with in collaboration with Adriana from the Institute Adolfo Lutz about the circulation of these totorivirus and other emergent enteroviruses that were introduced in Brazil. At least it started circulating the last decade, and nowadays are a common problem in terms of the swine herds. And another worry that we have is about the spillback. As I said before, like we have sensed over the years, opossums and other species are prone to receive viruses from humans. And we saw this is not uncommon, unfortunately, in Brazil. In another work with these opossums, what we saw is that they were infected in different waves of SARS-CoV-2. They were infected by different lineages of SARS-CoV-2 that were circulating during the pandemics. And it is unlikely that these animals have really participated in the transmission chain. We know that it is very possible that that fortunately these were non-productive infections, but it is quite remarkable that we were able to find genomes of SARS-CoV-2 in these animals pertaining from different lineages with specific mutations after they were infected. In Brazil, another species that were hugely impacted by SARS-CoV-2 were the coatis. There is a group from the Federal University of Minas Gerais that saw that these animals were actually infected more than one time by SARS-CoV-2 as well. We do not have, at least already, a situation that is close to this of the white-tailed deer that you have in the United States. We never saw the continued infection and transmission of SARS-CoV-2 in other species in Brazil, but we have to be vigilant for this matter. Another type of work that we are doing is whether even arboviruses could be reaching animal species in Brazil. And actually, Leila is doing a very interesting work about the possible introduction of Oropushi in different animals, especially in tapirs and anteaters and armadillos as well. And we are also looking for different type of emerging viruses arboviruses that are infecting and causing important disease in horses in Brazil. This is also part of our work. Going for the final remarks, we are also doing a lot of work in what regards to the surveillance of viruses in sewage waters, and we saw not only SARS-CoV-2 over time, but also different types of hepatitis viruses, IH viruses. These works are being coordinated mostly by Paula Hall at the University of the State of São Paulo in São José do Rio Preto, but we have a lot of works being conducted in that same matter in different parts of the country. And what we saw is actually that sometimes we see viruses that we do not know that were already circulating in human beings, mostly because of the lack of diagnosis, especially for gastroenteritis. But in the end, we saw these viruses circulating in water, and we could correlate whether they were causing localized outbreaks or not. Another type of work that is also in the scope of the network is about the continued monitoring of the circulation of different respiratory viruses like SARS-CoV-2. This work was a work from the group of Mauricio Lacerda Nogueira, showing the whole circulation of different SARS-CoV-2 lineages over time in São Paulo. And we are proudly also part of the efforts that the genomic characterization of dengue 2. This work was actually also coordinated by Mauricio Nogueira, in which they did the genomic characterization of dengue during the trial of the Brazilian dengue vaccine. That's nowadays a reality. It will be introduced very soon in the Brazilian health system, and this will be a landmark for the prevention of dengue, of course, in Brazil, and the network was part of this effort. And as a conclusion, other important stuff that we have here is about the monitoring of viruses that could be transmitted by foods. And I'm part of the roster of the FAO and WHO that is discussing this matter. And we saw this happening before here in the southern part of Brazil, including the possible exportation and importation of different viruses, sometimes by the lack of hygiene in the preparation of meat and all this that is commercialized in our borders. And the thing is that, well, viruses could be in our dishes, so we have to avoid, find ways to avoid that they could be there. And we have already, the FAO and the WHO have already published 2 volumes about this matter that are very interesting, and I recommend that you could assess them and read the remarks that we have in this group. Well, all that work that was presented to you is sponsored by different agencies. The INCT-One is especially funded by the Brazilian National Research Council, the local state agency for research, which is called FAPERGS, but we have support from many different other agencies, and we try to work in very close collaboration with the Ministry of Health and many other authorities in Brazil. And we, as I said before, we are prone to collaborate and to look for international partners in our network. Thank you very much for this opportunity. I'm open for the Q&A, and here you have not only my email, but you have also the Instagram link of our institute, in which we normally share the information that is being raised by our activities. Thank you very much.
Fernando, this was absolutely terrific. I think you've got, 50, 60 people salivating about all of the extraordinary opportunities that you've taken advantage of and you are continuing to provide leadership of. Let me, as questions come in, let me see if there are any questions yet. Yes, there is a question already, so I don't have to start out. I'll save mine for later. So Dr. Gu asks, my main research focuses on influenza viruses. So I'm very interested in the current situation, highly pathogenic H5N1 avian influenza in Brazil, especially status in cattle and domestic poultry. Are there any related epidemiologic investigations or virologic and serological studies? Thank you.
Well, that's a great question. Thank you very much, Dr. Gu. The thing is that it is a complicated matter in Brazil because H5N1 is on the scope of the activities of the Brazilian Ministry of Agriculture in what regards of the presence of the virus in domestic animals. We, as researchers, we are allowed to detect and characterize the virus in wildlife, but every time that you see an outbreak of H5N1, it could be backyard poultry or industrial husbandry. The thing is that we are not allowed to be there. This diagnosis has to be done by the Ministry of Agriculture. They do very, very comprehensive works in terms of the searching of the viruses, normally by PCR and in poultry herds, and they normally gives reports of it from time to time. We had an outbreak in chicken last year here in Rio Grande do Sul, and they did a genomic characterization of the virus that was circulating in that outbreak. It was very interesting that the genomes that were published are quite close to another outbreak that occurred at the same time in Azul, that is located like 50 kilometers far from the focus of the disease that was found in chicken. So they never reassured that, but it seems that there was a connection between these viruses. This year, there was a major outbreak, another outbreak of H5N1 in backyard poultry in Mato Grosso do Sul, which is in the southern western part of the country. And it is interesting that the ministry was aware that this was a second introduction. The first one was an outbreak caused by one of the lineages of H5N1 that was already circulating in wildlife in Brazil, and the other one seems to be another lineage. It could be transmitted had to be transmitted by another wildlife species, or even it could be another problem with the transmission between borders with other countries from Latin America, but we do not have any deep information on that. We really depend on this, on the information that is brought by the Ministry of Agriculture. They do very comprehensive surveys, as I said, based mostly in genome detection, but we do not have any other information that is available in what regards to serological surveys, for example. And I would say that chicken are their targets. We do not know about any organized surveys that are being done in cattle, for example. And every time that we ask them, they normally answer that cattle there is no report of cattle infections by H5N1 in Brazil, so this is not important until now. Some of our groups have did we tried to look for the hits of the genome in milk, for example, and but until now we never saw any possible positive detections of H5N1 in Brazilian milk. But of course, we have to be aware that this could happen because the amount the amount of Brazilian dairy farms is still huge, and sometimes they are geographically very close to the places in which the wildlife could be using as nesting sites, or even to poultry farms. So it's quite common that this is the scenario in Brazil.
Thank you. Thank you. We have another question from Christine Carrington in Trinidad. Thank you for a nice presentation. Very nice presentation. She says, we have been screening bats in Trinidad. We have several alpha coronavirus RDRP PCR positive samples and can recover RDRP sequences using metagenomic sequencing. But we have not so far been able to recover spike sequences. Any advice?
Christine, is actually amazing that we are asking this, because I had I think that it was 3 weeks ago. I was chatting with people from the UK, from Fulbright Institute, and they told me that there are people seeing the same as you in Trinidad. They are seeing a lot of different alpha coronaviruses circulating in bats, but unfortunately, they are not getting the spike sequences when they use metagenomics. What I can tell is that it is not easy. I unfortunately, I have to agree with you. Sometimes we have the whole RD and RP, sometimes the whole ORF1AB of these viruses, but it it is quite difficult to have the spike sequences. What we are trying to do right now is to do some sort of genome walking, trying to go further with conventional PCR and sequencing, but it's not easy. We already tried many protocols, include these which is now not available anymore, but this pan coronavirus that was a kit that was until last year. And the thing is that sometimes we have only partial sequences from spike of these viruses. It is not very easy. What we are trying right now is to isolate these viruses in different types of mammal and bat cells to check whether we'll be able to have them in greater loads to try the sequencing, but it is quite challenging. I would love to have the opportunity to have contact with you, and maybe we could share some ideas on this, but it is not that easy, actually.
Thank you. And Dr. Gu says again, although there have been no reported infections in cattle, given the current situation in North America, would proactive H5N1 surveillance be necessary?
I would say so. I totally agree that it would be necessary. One of the things that I think it would be very important is that we could be allowed at least to do serological surveys to understand whether the virus has or not circulating, or it was circulating in animals. But unfortunately, we were not allowed sometimes even to import the regions to do so. So we we have our hands sometimes tied to do that type of effort, but it would be important. And in many times when we have the chance to chat with people that is in charge of this type of surveillance at the Ministry of Agriculture, we tell them that it would be important, even to share the information that the virus is fortunately not circulating there, but as you said, based on the experience of the United States, we have to do something about that, at least to monitor the presence of the viruses.
Wonderful. Dr. Piedade, João Piedade, he asks, is there any investigation on arenaviruses and their natural hosts going on in Brazil?
That's excellent question. Actually, we until now from the hundreds of samples that we have looked at from wildlife in our network, we did not see whether arenaviruses were there by metagenomics, and we have a lot of work being done in trying to detect this in different sorts of rodents by PCR, but luckily we are not seeing them in our work. But yes, there are many groups working on this, and particularly there is a worry about these emerging arenaviruses that are circulating, sometimes provoking important outbreaks, I would say, in Brazil. From the last years, we had this reemergence of two or three cases of Sabia virus, and this is one of our main worries. These are the types of viruses that could be really a menace to human health. We are now conducting a particular effort in the Amazon, looking for the possibility that we could find different arenaviruses in rodents there. But there are excellent work being done by different groups on what regards to arenaviruses. Inside our particular group, what is being done is with arenaviruses, the effort that is being done by Rafael Elias at the CNPM, which is the Brazilian National Institute for characterization of protein structure and all this. And Rafael is now working with the structure of the viruses, of the viral proteins, and there are also work being done by different groups. And of course, arenaviruses have to be in our agenda due to the amount of arenas that have already been described in Brazil and in Latin America as well.
Thank you. Excellent. I have a question, which is that sometimes structural elements are increasing risk, such as the deforestation of the Amazon. And there's been a tremendous political divide between individuals political leaders who encourage deforestation and colonization of the Amazon versus those who are more conservation oriented. In the United States, we can definitely relate to this because we have exactly the same divide, political divide between persons more focused on conservation and persons more focused on development. Would you just comment on where we are in Brazil in terms of public attitude and political policy around the Amazon jungle?
Well, this is also part of the so-called polarization of politics that we have in the world. And in Brazil, it's not very different. Actually, what we had in the last, let's say, 10 years is that the Amazon was hugely impacted, at least from 2015 to 2021, 22. The amount of deforestation in the Brazilian Amazon was very huge. And actually, it was, of course, a threat for conservation of species, and it could be a main factor, as you said, for emerging of different viruses and pathogens. And we had this worry, and it was colored by politics, of course. But there are other minor points that have to be addressed on this. And two of them is that Brazil is a federal, has a federal organization in which states have not the same amount of power that states have in United States of America, but they have some power. Sometimes local governments could act in a way that is somehow different from what the central government could have. We have nowadays a very strong woman in our Ministry of Environment, Marina Silva. She is very prone to defend the will of the people from the forest and all this. She's very acknowledged by that. But the thing is that sometimes all their interests come closer. And this is another point, apart from the state organization, is that sometimes even for the federal government, it's kind of difficult to hold the will, for example, of the petroleum industry. And there is a huge interest of different oil companies to be there in Amazon, at least to check whether there are some important reserves of oil underground. Another interest that is driving some changes in despite of the will of conservators of the forest is that road connections are very important in Brazil, because we lost all other types of train connections between states and all this. And there is one main road being planned and being actually, it is at the beginning of the construction of this road. And this will like cut part of the Amazon in two. And of course, every time that we have a road, you're gonna have further urbanization, industry coming in the next year. So this is another worry that we have, summed with the deforestation that we had in the last decade that is now diminished or lowering, let's say. In the meantime, we think that at least from the southern part of Amazon, it could happen again because of the construction of this road that will connect the far portions of the Amazon to the center of the country. And that is worrying because it will pass through very conserved points of the Amazon that were not touched by the humankind already. And that will happen probably very soon in the next few years. And that's worrying. That's the situation right now.
Thank you. That gives us a lot of context. I think we have one final question from Professor Billy Hall in Ireland. Have you any evidence of bat to human transmission using serological methods, for example?
Well, I think that the audience have guessed many of our further moves. And the idea is that now we are trying to not only isolate some of these viruses in vitro, but we will also we are also planning to have their spikes and other possible serological targets cloned. And the idea is to do some serological analysis about the possible circulation of these diverse coronaviruses, not only in humans, but we are quite interested in the circulation of these viruses in different types of animals, and particularly in domestic animals like cattle, swine, are in our plans to check whether these animals have already some sort of history of contact with these viruses that we are seeing, especially in bats.
Very good. Well, Professor Spilki, we've been very lucky to spend an hour with you, and we're very grateful for you contributing to this webinar of the Global Virus Network. And we'll let you get on with your work day and congratulate you on your fine progress in your career. It's inspiring to hear about your work and the broad vision that you have as a One Health leader on our hemisphere. Thank you again.
Thank you very much for the gentle words, and thank you very much for this opportunity. And as I said before, we are prone here to receive questions and possibilities for collaboration with groups from abroad. So thank you very much for the opportunity anyway.
All right. Bye-bye.
Bye-bye.